Sanibonani bafundi and namkelekile to another exciting life sciences lesson. Now, all living organisms live in environments that change. The internal environment of the living organism is also changing. Therefore, if all living organisms are to survive, they must make changes to their body to adjust to the changing environment, both inside and outside their bodies. They aim to maintain as balanced a condition as possible. This is known as homeostasis. Now, you'll learn more about this condition and the processes that take place to maintain homeostasis over the next few lessons. But in order to keep making checks and keeping balanced, the organism has to be able to sense what is happening in the environment. Sensitivity is the ability to detect change and then to respond to it. Any change that can be detected and causes a change in the body is called a stimulus. The cell or organ that detects a stimulus is called a receptor. The change that takes place in the body is called the response. The part of the body that responds to the stimulus is called an effector. Now, all living organisms must respond to stimuli. Plants are no exceptions. Now, in this lesson, we'll look at ways in which plants respond to their environment. Plants cannot respond to a stimuli by moving their whole bodies as animals do. Instead, different parts of the plant change their growth in response to stimuli. For example, shoots grow so that they bend towards the light. Roots grow down into the soil. Flowers grow during the seasons that are suitable for seed production. Buds and seeds may be dormant during unfavorable seasons such as cold or drought and then grow when conditions improve. Now, chemical substances called plant hormones cause many of the plant's responses to the environment. Plant hormones affect growth and development of plants. Only very low concentrations, usually below 1 part per billion parts, of plant hormones are needed to have these effects. Plant hormones may be produced in the same tissue where they cause a response, or they may act on tissues that are some distance from the place where they were produced. Now, who discovered plant hormones? Now, you might be surprised to learn that the same man that revolutionized thinking about origins of life, Charles Darwin, was one of the first scientists to explore the responses of plants to external stimuli. In 1881, Charles Darwin and his son Francis did experiments on calyptiles, which are sheaths that enclose young leaves in germinating grass seedlings. The Darwins discovered that when they exposed calyptiles to light from one side, the calyptiles bent towards the light. They removed the tip by cutting just the very tip of the calyptile and then exposed the calyptile to light. Nothing happened. They covered the tips of the calyptiles with metal foil and exposed them to light from one side. The calyptiles grew vertically and were very apparently unaffected by the light. They removed the metal foil caps. The calyptiles bent towards the light again. The Darwins also noticed that each calyptile curved a short distance away from its tip. The very end of the calyptile was not the area that bent. Slightly behind the very tip was the area that bent. So, what did the Darwins deduce from the experiments? The Darwins suggested that the receptor was the light-sensitive region at the tip of the calyptile. The effector was the growth effect, which happened a short distance below the tip where the calyptile bent towards the light. A substance moved from the calyptile tip to the cells lower down, where it caused a growth response. Now, in 1928, a Dutch plant psychologist, Frits Wendt, continued with this research. He extracted the growth substance from the calyptile tips. He called it auxin. Several auxins have been found in plants. The most well-known auxin is called indole acetic acid, or IAA. Auxins are produced in the meristems or growing tips of the plants. Auxin moves into other tissues. It causes young cells to elongate. The young cells can elongate because they take in more water, and their cell walls are still elastic to allow for this growth because they are young cells. Auxins do not cause all young cells to elongate equally. In shoots, the more auxin that is present, the greater the elongation of the cells. Lateral buds are less affected by the auxin than main shoots. In roots, auxins only cause elongation at very low concentrations. At higher concentrations, the cells lengthen less than they do with pure water. Now, after the break, we'll look at the effects. I'll see you right after the break.